Alistair is a plant pathologist and mycologist at the University of Queensland. He brings his expertise in Myrtle Rust to the Beyond Myrtle Rust research program, and he focuses on fungal genetics. I'm certainly looking forward to hearing more about that today. And Alistair, are you all good to go there? I can certainly see your screen.
Yeah, ready to boogie. Thanks,
I think
Renee.
you can take it away.
Yeah, I know that you're looking forward to this, but I can tell you it'll be soporific. So, yeah, hey everyone, I'm Alistair. Thanks for joining us. Today, much like myself and my teenage friends, we are going to be talking about hypothetical sexual reproduction in Austropuccinia psidii. The question being whether different strains might outcross on a universal host. Hopefully, it's something that will always be hypothetical and we'll never actually know the answer to. I'm going to take you through the hypothesis that we tested and how we tested it, and then you can make up your mind at the end as to whether you think they outcross. This work, or all this research, was in collaboration with Jessica from Brazil, Louise. Oh man, I can't see who else is there. Beck, and probably Perry. Sorry, the little screen is blocking my screen. All right, pretty much if you've heard me explain a rust life cycle and lost count of all the times I've explained it, tune out for 5. This is just background for me to set up why myrtle rust does reproduce sexually. So rusts, well, I should say plant pathologists and mycologists, when we think of a rust life cycle, we think of Puccinia graminis, the life cycle of wheat rust. And rust fungi are so successful because of this clonal spore stage, uridinospores. Compared to other Basidiomycota, rust fungi having that clonal stage is such an advantage. If you think about smuts, their clonal stage is non-pathogenic, and a lot of Agarica and Mycotina won't be pathogenic in their clonal stage or have decreased pathogenicity. So this clonal stage in rust fungi that's pathogenic is just so killer. So at some point in time, that fungal hymenium gets a trigger to produce telia. We don't know what that trigger is either. It could be leaf senescence, it could be a change in climate, it could be a signal from the rust or a signal from the host. And that's when you've got this dikaryotic spore stage where you now have a fusion of nuclei, karyogamy, followed by eventually meiosis, which produces haploid basidiospores. And then in Puccinia graminis, because you have an alternation of hosts, it's a heteroecious rust, these basidiospores infect barberry. So these are produced on wheat and these are produced on barberry. These basidiospores infect barberry and produce a spermagonium if they cause an infection. And a spermagonium or spermagonia kind of amplify the haplotype from the basidiospores, and they produce hundreds to thousands of little spermatiospores, all with the same haplotype. And these can cross-fertilize other spermagonia to form a dikaryon again. And in wheat rust, this stage, the aciospores, the acea, they're making spores to get you back to the wheat host. So pretty neat. So you can imagine, because this rust life cycle is so well known, we try and apply it to every rust, and there are about 10,000 described names in rust fungi. With myrtle rust, you can kind of picture it came from Brazil in a tropical area on evergreen hosts. So it's possible it doesn't really need stages that let you change hosts, and it doesn't really need stages to overwinter. Myrtle rust has uridinia, these bright yellow things, and telia. And those teliospores, when you give them a chance, they germinate quite readily to form basidiospores. So uridinospores, teliospores, and basidiospores. And the early community, there was some ambiguity about the life cycles because there was no spermagonia and acea. And one of the hypotheses, and it's still circulating today, it's still, people are recalcitrant to accept that there's sexual reproduction. So there's a hypothesis still floating around that myrtle rust is strictly clonal. And so we first tested this hypothesis. Sorry, you've probably seen this slide a million times as well. We just separated the gametic stage, basidiospores, from the mitotic stage, uridinospores. We put those basidiospores onto emerging leaves of Syzygium jambos, and we essentially saw what we would expect from a dihybrid cross. So we saw a random inheritance of alleles that we knew were heterozygous in the parent. So the F1 progeny had a random assortment of alleles, which is really good evidence of sexual reproduction. And sexual reproduction makes so much sense in the endemic population of Myrtle Rust, or Austropuccinia psidii, in Brazil. So this is just a splits tree network of multi-locus genotypes from microsatellite data of a population of Austropuccinia psidii on eucalyptus in Brazil. The paper is Grasser et al., I think 2013. And what we see here is really great evidence of the clonal stage doing its job. So lots and lots of multi-locus genotypes, one multi-locus genotype that's shared by lots of single uridinial pustules, same here. And the rest of this high genotypic diversity is best explained by sexual reproduction. And what's cool about a splits tree network is that it shows every possible hypothetical relationship among those microsatellite data, or among any data when you use a splits tree. And the more articulation you see, the more kind of evidence or support you have for reproduction. Homoplasy being the other potential explainer for why there's reticulation. So yeah, the point of all that is that Austropuccinia psidii reproduces sexually. So many moons ago, in fact about 4 years ago, we hypothesized that this was the life cycle, nothing too dramatic, no spermagonia produced, no asea produced. And the gray area in the life cycle of Myrtle Rust is kind of what happens on the leaf after these basidiospores meet each other. So right here is a picture that Stuart Fraser and I took ages ago. I can't see it myself, but it's got basidiospores, and you'll see that they each have 2 nuclei. And so our hypothesis is that they're homokaryotic, and this is just based on the life cycles of other rusts. So after that after meiosis, you have a mitotic division that produces 2 nuclei that are exactly the same. And basidiospores germinate at these polar ends. And so our hypothesis is that those 2 nuclei migrate down those 2 germ tubes and potentially could cross. So that same haplotype could end up in 2 genotypes based on that. Now that's never been published. It's always been made to be taken out of any manuscript that we've put forward with that hypothesis. But I think that's probably what happens. So it's still a gray area. It's not been published officially, but we're pretty confident that that's what happens based on what we've observed and based on what's seen in other rusts. If you want to check out another example of a rust, Puccinia alleli has 2 mitotic divisions. So each basidiospore ends up with 4 nuclei that are all homokaryotic. Okay, this slide is a PCA, principal components analysis. It's just clustered like multi-locus genotypes, and they're colored by the origin of the population. So for the last 50, 60, 70 years, like since the 30s, I think, the Myrtle Rust is known to have some host specificity. And that's when people were experimenting by putting uridinospores from guava onto eucalyptus, and eucalyptus uridinospores onto guava, and not getting these cross infections. And it was really conclusive in that Grasser paper from 2013, I think, that the native populations of Myrtle Rust in Brazil were structured by host. So a guava, the rust on guava never outcrossed with jabuticaba or pitanga. And now we're in a little bit of a jam because we've introduced hosts to Brazil that kind of accept all these strains like Syzygium jambos, and we've got these mobile strains, the South African strain and the pandemic strain, that don't really have a defined host range. So they're incredibly polyphagous. So where a basidiospore, when populations were structured by host, a basidiospore from guava might not be able to infect jabuticaba, but now both of these might be able to infect Syzygium jambos and physically cross. So that's the background. Myrtle Rust reproduces sexually, and now that we've got these universal hosts, can it reproduce sexually on one of those hosts now that they're not structured by host range. So those are the big questions from resolving the life cycle. And everything from here on is now new. And this is where it gets really boring. This is the first time I've ever explained it. This hasn't even, I don't think it's been submitted yet. So bear with me. If I make things even more confusing, then check out this really cool chapter by Colo et al, Fungal Sex, the Basidiomycota, which is where this figure is adapted from. So I'm just going to take you through the types of mating in Basidiomycota and then the loci that we're dealing with. So in fact, I'll just get rid of the loci right now. So you've got two loci that you're always going to hear me talk about: the homeodomain locus, which has homeodomain genes, and the pheromone receptor locus, which in smuts has PRA genes and in rusts has these STE3.2 genes. And in other Puccinia it has STE3 genes. So in bipolar mating, it's comparable to humans with an X and a Y chromosome. So your compatibility for sexual reproduction is controlled at one locus. And the example here is sugarcane smut, Sporosorium scidominium. So on this chromosome, only separated by 50,000 base pairs, you've got the homeodomain locus and the pheromone receptor locus, and they're linked in recombination. So one of the things about controlling mating compatibility is that the loci have to differ. So something must differ between MAT1 and MAT2, and these MAT1 and MAT2s are not the same as MAT11 and MAT12 in Ascomycota. This is just mating type. Gosh, did that get confusing? So bipolar mating in Sporosorium scidominium, even though there's two loci, they're linked. So it's controlled, there's genes at a homeodomain locus and genes associated with pheromone receptor locus, but they're linked in one locus. So that's bipolar. You can think of another even more simple example is Eustilago hordii, which has just lost the pheromone receptor locus. So it only has homeodomain genes, and that's a bipolar mating strategy. Maybe this is going to make more sense because I feel like I butchered that when we look at what a tetrapolar example is. And our tetrapolar example is Microsarcoma matus, corn smut. And so immediately you'll see that one of these, the homeodomain locus is on chromosome 1 and the pheromone receptor locus is on chromosome 5. So they're definitely not inherited together in meiosis. You can have any combination of these. Whereas in Sporosorium scidominium, these are inherited together in recombination. So yeah, the pheromone locus must differ and the mating locus must differ for a compatible cross in a tetrapolar mating system. So what do we know about this in rust fungi? Well, the first rust that these genes were kind of found is Puccinia tridacina, and they had a homeodomain locus with 2 HD genes, and then they had these 3 homologs of a pheromone receptor locus with STE3 genes. And what was confusing was that there was 3 of them. They didn't really know where they were in the chromosomes because the assembly at the time was not highly contiguous like it is now. So you'll just see question marks everywhere. So they weren't sure if it was bipolar or tetrapolar or what was really going on. And just in August this year, they phased the genome of Puccinia tridacina, and they say that it's a tetrapolar mating system because HD, the homeodomain locus, and the STE3 homologs are on separate chromosomes. Now, I think let's come back to this. So they concluded it was tetrapolar because it's at 2 different loci. But I think, yeah, I'm going to talk about this in a second. Let's just keep it in the back of your brain tanks. So that slide, complicated explanation, HD locus, pheromone receptor locus must differ in tetrapolar mating, and for any compatibility, they must differ even in bipolar mating, and tetrapolar and bipolar are the 2 mating types in Basidiomycota. So when we look at these genes in Myrtle Rust, we looked at the genomes available for the pandemic strain, the South African strain, and a strain on eucalyptus in Brazil. And we just pulled out the homeodomain genes and the STE3 genes at the pheromone receptor locus. And with the homeodomain genes, what we saw is that each genome had 2 alleles for each gene. So 6 copies in toto from those 3 genomes for HD1 and 6 copies in total for HD2. So highly variable and allelic. When we look at STE3, at the 3 copies of STE3, this branch length gives it away. And I should say that these are translated genes. So Austropuccinia psidii, totally identical in STE3, STE3.2.1, 2.4, where is it? Down here and down here. Okay, so fully identical. Oop, I don't know if I was finished. Somehow that I must have used an error. Basically, no variation in the translated amino acids of those STE3 genes among those 3 different strains. And then when we take a look at the locus itself, this is the most variable of the 3 STE3 genes in the pheromone receptor loci. So the variation, this yellow thing is the translated gene, or the kind of the gene map. You'll see that the SNPs, there's a couple of SNPs in exons here and here, but most of the variation is flanking the actual gene itself, or is in intronic regions like this. If there was a SNP in the exon, then we already know based on the translation that it must be a synonymous substitution. So almost no variation in those pheromone receptor loci across those 3 homologs of STE 3.2. So over 8,000, almost 7,000 and 4,000 nucleotides, you have greater than 99% pairwise identity and absolutely no variation in the translated amino acid. Contrast that with the homeodomain locus, which is really variable. Now, what does all that mean? Well, basically, STE 3.2 is not involved in mate compatibility. So it still might be functional in recognition, pheromone recepting, but it's not controlling compatibility. So the basic principle is that they must differ for there to be a cross. And this has happened in other Basidiomycota. So we take STE 3.2 out of the equation, and here's that hypothetical situation. So we've got 4 basidiospores here. Each has a different set of alleles at the HD1 locus. Here we've got HD1 has a big A, HD1 has a little a, HD2 has a big B, HD2 has a little b. These 2 are compatible because they differ at each HD gene at the HD locus. When we try and cross it with this basidiospore though, this basidiospore can't cross with this one because the HD1 gene has the same allele and the HD2 gene up here has the same allele. So this one wouldn't cross with these 2. The big question, so we're au fait with that. That's pretty understandable. This is where it gets hypothetical. So we could pretend that this one's the pandemic strain in New Zealand and Australia, and down here's the South African strain. So we've got C's and D's at our HD locus instead of A's and B's now. Something that's been separated by their host range for, you know, a couple of 100,000 years. Will these homeodomain alleles be able to outcross with these homeodomain alleles? And that's where the question mark is. The reason that we say yes is that variation at that homeodomain locus drives compatibility. Pretty much all you need to be is different to be able to cross, and then back that up with totally identical pheromone receptor loci if there is a pheromone, if there is a function for the receptor there. So they will recognize each other, and then they'll be compatible at this HD1 and HD at the homeodomain locus. But hopefully we'll never find out if we're right or wrong, but we hypothesize yes, they will be. But yeah, big question mark there. Okay, that's a lot of content. And this is all pretty boring. In fact, I encourage you to read the Colo paper if you want to hear about this. This is pretty much the long and short of it is, why would Myrtle Rust become bipolar if it was once tetrapolar? Bipolar mating just increases your chances of sexual reproduction. tetrapolar mating decreases your chances of inbreeding, but in a way it also decreases chances of sexual reproduction. And in taxa that have the gametic stage getting distributed along long distances, you know, inbreeding might not be a problem. Okay, so in that Colo paper, check this out. This is their wording, not mine. You can tell because of the nouns. So they say the transition to bipolarity through loss of type determination in pheromone receptor genes, which is saying that it's happened in lots of Basidiomycota, that pheromone receptor gene has gradually homogenized. You end up not having any variability at that gene, and you essentially become a bipolar. Your mating system becomes bipolar. So maybe at some point in time, Austropuccinia psidii had a tetrapolar origin because the loci are totally unlinked, but it's lost any variation at the pheromone receptor locus. And so you would consider it bipolar. This is where we can link back to that Puccinia tridacina study that concluded it was tetrapolar. Probably they want to look a little bit more at the population to look at the variation in the STE3 genes, just to check that what they're looking at is a true tetrapolar fungus rather than something that's also gone to bipolar. It's going to be super interesting in rusts just to see how it all plays out with bipolar versus tetrapolar mating. All right, all that work was part of a study in Brazil where they were looking at the production of telia, and they said, does this make sense? And I was kind of like, oh, well, you want to look at sexual reproduction, let's go look at these mating genes. But what Jessica had seen in a time course study was that telia production on Eucalyptus grandis increased over time, and there was an upregulation of meiotic genes, as you would expect with increased production of telia. So it just seemed like a good opportunity for me to slip some mating loci stuff into someone else's study. Yep, so key findings, Austropuccinia psidii, it's got all the equipment, genetic equipment for sexual reproduction. The ST 3.2 homologs are near identical, and HD loci are variable. That identical at the ST3 genes is really good indication it's bipolar. And then this is up to you to decide whether I've convinced you or not, whether basidiospores from different strains that are now no longer structured by a host can outcross on a shared host. We think yes, because of that variability at the HD locus. And that, Renee, is me done.
That was intense and fantastic. Thank you very much. I'm not sure how much I can contribute to this particular question time. So if audience members, you do have questions, please get them in the question box. Really the main thing I was thinking about with regards to all that and kind of moving it beyond the theoretical stage is next steps in terms of actually testing. I suspect there's some big logistical challenges there though, in terms of it not being ideal to move new rust strains between different countries to get some things tested on plants.
Yeah, exactamundo. Yeah, we don't ever want to know. Possibly you could test it in Brazil with like Jabuticaba and the eucalyptus one, but I mean, why would you intentionally make new crosses? Probably not a good idea. But yeah, I don't know. I wouldn't expect any questions. I still have questions myself about it.
Well, I do want to reassure you, Alistair, that you did not lose your audience, at least. And I know these webinars can be a bit disconcerting in terms of not being able to see all of the faces like you do on Zoom or something like that. So you did an excellent job there of sprinkling humor throughout and keeping people with you, because we did have and keep around 55 audience members for that entire presentation. Okay. All right, here's a good question. What are your key questions, Alistair?
Oh,
fair
bless.
Point.
What are my key questions? Well, I answered those. Really, they were, is it bipolar or tetrapolar? And will they outcross? Hopefully we never ever know, but I think that they will. So yeah, that is the key question ever since we resolved, or we think we resolved the life cycle. I guess the jury's still out on whether we did or not. But yeah, those are the big standing questions, is can there be outcrossing of different strains?
All right, well, seeing as, oh, just in time, someone's got one in here. Right, in the figure, should be sharing these with the audience. Let me see. Everyone can wrap their brains around this alongside me. Right, in the figure with all the other countries, there are isolates from Argentina, Paraguay, Colombia, etc., but not Uruguay, which, like Brazil, is part of the natural range of this pathogen. Any reason why Uruguay is missing? Is any work being done on obtaining isolates from there?
Well, I guess the reason is because they're not, I don't have them in my data set. I'm sure that someone's got them somewhere. So I can only work with publicly available data or data that we've acquired. And I think I've made this data set publicly available. Probably it's on my ResearchGate thing. So if you've got data from Uruguay, then you can pop it in. It's just at those six microsatellite loci. That's all that's behind this PCA. So you can pop it in, you'll work it out, and we can make a new, what's it called, PCA, if you're keen.
You certainly could. I think there's probably some colours left to spare there. A couple more good questions now coming in. So what are the implications for Australia and New Zealand in trying to manage myrtle rust or keep new strains out?
Yeah, it would just be, I guess now that we're kind of living with myrtle rust, the worst thing that can happen is that we get in new strains, and then there's either they have a different levels of virulence on our currently non-susceptible hosts, or they outcross with the pandemic strain that's currently here, and they overcome any resistance that's present in the population. I think keeping out new genetic genotypes is one of the best things we can do now that we're living with myrtle rust.
It would certainly seem like a good idea. And so on that note, should there be...
Sorry, I think the next one is that South African one. So that's a flight from Joburg to Perth. And then once it's in Australia, you guys know you're going to get it. So it's
probably.
Yeah, it's a bummer.
There's certainly precedent for that.
Yeah.
Right, another question here. Should there be an expanded program of testing then, not to determine sexual reproduction, but rather to determine how many shared hosts there might be between some of the strains?
Well, I guess the host ranges of the strains, that's pretty much for wherever the strains occur. So for example, the South African one, it's just so embarrassing. Oh, shit. Oh no. Oh, I forgot.
Well, I think
their name. Oh, I'm so sorry.
It happens to us all.
Help me out. Works for Scion from New Caledonia.
Oh, Julia Suato.
Julia, sorry, Julia.
Fabulous previous presenter on this very webinar series as well.
Yeah, Julia went to South Africa and she screened a whole bunch of New Zealand plants against the South African strain, and I guess it's people who are doing that kind of great work that's gonna show us where the strains might also be able to outcross as well.
Yes, well, that's kind of point one, isn't it? It needs to actually be on that host.
Yeah.
To meet a fellow spore on the same host.
Oh, well, they can meet on any universal host. That's the problem. That's right. We've got so much of that around. Jamboss.
That's right. Well,
sorry, Julia,
yes.
forgetting your name just now. Oh man.
I know Julia well enough to know she's not going to mind Alistair.
Okay.
And she's just going to be very pleased to have had a mention in this webinar, I would think. All right, well, we don't have any more questions from the audience, so I think I could probably wrap it up there and let you get on with your day, Alistair, which is, of course, just getting started over there in Australia. Thank you so much. And a video of today's webinar will be made available on the Beyond Myrtle Rust website in the next couple of days, and it will also be emailed to everybody who registered for today's presentation. Now, the next one is going to be Wednesday, November 10th. We're going to be hearing from Karen Vandewalt, a conservation and science advisor at the Otari Native Botanic Garden in Wellington. We're going to take quite a different direction here because she has expertise in seed banking, and she has spent several years now researching this technique for Myrtaceae as part of a PhD project that she's just finishing up. Her talk title is Freezing for the Future. Can seed banking fleshy Myrtaceae seeds buy us time in the Myrtle Rust battle? So I know she's had some real challenges with these seeds. There's going to be some very interesting information in that webinar of real relevance to steps we might or might not be able to take in the face of managing Myrtle Rust in New Zealand. So please do join us. The invite should be in your inbox in the next couple of weeks. Until then, ka kite, and I'll see you all later.